Hey biologists! In this set of notes we are going to be learning about the history behind the race to discover the structure and shape of the DNA molecule. We're going to be learning about essentially the structure of DNA and its function. We'll be using a lot of terms such as nucleotide, phosphate group, deoxyribose sugar, nitrogen-containing base, covalent bond, and then we'll also be talking about purines, pyrimidines, 5' 3' antiparallel. And you'll also be learning about the scientists including Chargaff, Watson and Crick, and Rosalind Franklin. So the experiments that we've learned about so far, which were conducted by Frederick Griffith, the Avery McCarty-McLeod team, and Hershey Chase, identified that the molecule DNA, not protein, is the molecule that stores all of the genetic information used by all living things. This was essentially summarized as the DNA theory of inheritance. And the confirmation of DNA as this molecule inspired a race to discover the structure of the DNA molecule itself. There were scientists researching this all over the world, but the two countries which had the leading scientists in this field was a competition between Linus Pauling of the United States and then Watson and Crick and Rosalind Franklin and Maurice Wilkins in Britain. So we'll be talking a little bit about that through this. So we'll essentially be answering the question, how exactly does the structure of the DNA molecule enable it to store genetic information and allow it to be passed on from generation to generation? So let's start by thinking about the basics. So what do we know today about the molecule of DNA? Well, DNA is a type of nucleic acid. Remember we learned about the four major groups of organic compounds: carbohydrates, proteins, nucleic acids, and lipids. So nucleic acid is what DNA is an example of. And its name comes from the fact that within this DNA molecule, it also has a special type of a sugar called a deoxyribose sugar. So the full name of DNA actually represents its nucleic acid and structure. So it's a deoxyribo, hearkening to that sugar molecule, and then it's a nucleic acid. So deoxyribo nucleic acid is DNA. DNA is made up of five really important elements: carbon, hydrogen, nitrogen, oxygen, and phosphorus, or CHNOP for short. And it's that phosphorus element that is particularly important here. And if you remember back to the experiments conducted by Hershey Chase, they used the presence of that phosphorus element as a key distinguishing characteristic when they were conducting their chemical analyses to discover whether it was protein or DNA that had been injected into the bacterial cells by those viruses. So that phosphorus is very important. Here you can see an atomic breakdown, or kind of atomic structure of this molecule. We won't be looking at this particular model very often because it's kind of complex because you haven't taken chemistry yet, but I thought it was at least important to show you the relative balance of the different types of atoms present and the elements within this molecule. But we'll be looking at more simple forms of this diagram coming up. Oh, here's an example actually. So you've probably seen this double structure or double helix structure before, where you have two twisted strings and then things bonded together in the middle. And if we were to kind of untwist that shape and make it as a straight ladder, this is what we see here. So this is called the ladder molecule, and this is the helix molecule. And so this is a summarization of the atomic structure that is shown here. And we'll be looking at this particular part of the atomic structure coming up. So the monomer unit of DNA, or the small building blocks of the DNA molecule, are called nucleotides. And nucleotides were discovered by a scientist by the name of Phoebus Levene in the 1920s. So there was already some understanding, even way before the confirmation that DNA was the genetic molecule. So it's been known for a little while, but the scientists we're talking about basically expanded upon the research by Phoebus Levene. And each nucleotide is made up of three parts. So there's three parts of one nucleotide. There's one nitrogen-rich base. Okay, so there's a region of elements, or excuse me, a region of atoms that contain a lot of nitrogen. And this is the nitrogen base. This nitrogen base is bonded to a 5-carbon deoxyribose sugar. And we'll talk about the 5-carbon part of the sugar coming up, so don't worry about it right now. But just know that we've learned that sugars are a ring of carbons. So this is a closed ring of carbons. And so this is the deoxyribose sugar region of the nucleotide. And the last part of the nucleotide is the phosphate group. And a phosphate is a phosphorus element, or excuse me, phosphorus atom surrounded by oxygens. Okay, so this is your phosphate group. So please draw and label this in your notes. So pause the video. And the three parts of this nucleotide are held together by strong covalent bonds. So all of these solid lines represent strong covalent bonds that hold all of the three parts of one nucleotide together. And that's important because there's a different type of bond in the DNA molecule coming up that we'll be learning about. Let's go back to the fact that we talked about how this sugar is 5 carbons. So this is a 5-carbon sugar. The first carbon is here close to the nitrogen base. And so starting with this first carbon next to the nitrogen base, we have 1, 2, 3, 4, 5. And remember that carbon atoms are not always labeled. So we start with the base and then we end with the carbon close to this phosphate group. And this symbol here, this asterisk or this apostrophe, represents the word prime. So we call this 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So the 5 prime carbon of this sugar is close to the phosphate group. And so a memory clue, and this is important, okay, this is something you need to know. And so one way that you can remember where the 5 prime carbon of the sugar or of this molecule is I think of like if I'm standing, this is gonna be weird. If I'm standing and so if I'm standing and going like this, here's my high 5, my 5 prime carbon, so this is the phosphate group sticking up, and then my arm out here represents, so I'm the sugar, and my base is over here. So my nitrogen base is like this, and I'm giving you a high 5, and I have 5 fingers on my hand, and that's the 5 prime end of my molecule. This is important because it has to do with how these strands of the nucleotides are arranged within the whole molecule. So think of high 5 as the 5 prime end sticking up with the phosphate, okay? That's important for coming up. All right. Now so we've talked about the 5 prime carbon or 5 prime sugar. Let's talk a little bit more about the nitrogen bases. This is a really important part because it's the 4 different bases that essentially vary within the molecule. So this basic structure repeats throughout the whole molecule, but what changes is the base between 4 different types. So there are 4 different types of nitrogen bases in the DNA molecule, and they're arranged in 2 classes based on their structure. One class of these nitrogen bases are called pyrimidines. I'll say that again, pyrimidine. These pyrimidines are a smaller single ring of nitrogen. So we have 1 hexagon here of nitrogen. And the 2 nitrogen bases that are examples or subtypes of pyrimidine are cytosine and thymine. And the second class of nitrogen bases are called purines. They are a larger double ring of nitrogen, so we have a pentagon bonded to a hexagon, and these are adenine and guanine. To remember which are which, I have a memory clue for you. So if you think about all of the names of the 4 nitrogen bases, the pyrimidines have a Y in their name. So pyrimidines are cytosine and thymine. Therefore, guanine and adenine would be the other type. And in terms of remembering which of these is larger and which of these is smaller, I remembered that pyrimidine is the longer word, but it's the smaller molecule, so it has only 1 ring, whereas purines has double ring. That's a really kind of tricky memory clue. I don't have a better one for you, but longer word, smaller molecule is what helps me remember which of the 4 bases are pyrimidines because of the Y, and it's a smaller molecule because it's a bigger word. Opposite. Okay. All right, let's talk about the 4 nitrogen bases again. So the base pair rule was an important discovery by Erwin Chargaff in 1949. And he was conducting studies about the relative amounts of these nitrogen bases in a strand of DNA. So, for example, from a grain of wheat, he extracted the DNA molecule and he analyzed it to see how much of each of these bases was present in the DNA molecules within the wheat. And what he found is that approximately 27.3 of a DNA molecule contained adenine, and approximately 27.1% of that molecule was thymine. And then he compared G and C, and he found that 22.7% of wheat was G and 22.8% was C. So he recognized, when he did this comparison in other species, he found that there was always a comparable percentage in G and C and A and T. So hopefully you recognize this pattern here, which you might have also noticed if you did the DNA structure explo the other day. But basically the base pair rule can be summarized as there's relatively the same amount of A's as there are T's, and there's relatively the same amount of G's as there are C's. Furthermore, the amount of each base varies within each organism. Not every species has the exact same percentage, so it's a little bit variation. And this basically means, to summarize, that therefore an adenine will always be opposite from a thymine because they're found in equal concentrations. One would equal the other. And this also therefore means that the adenine, which is a purine, would bond to the pyrimidine, thymine. Same thing with G and C. G is the guanine, which is the purine, and cytosine is the pyrimidine. So the following rule is basically that you always have a purine paired with the pyrimidine. You always have A bonded to T and C bonded to G, unless a mutation has occurred. So let's do a little bit of problem solving to see if you can answer this question. Imagine you extracted some DNA from an organism and you did a chemical analysis to determine how much of that DNA molecule was A, T, C, and G. Say 36% of that DNA molecule was made up of adenine bases. Solve for the remainder of the bases. Pause the video and see if you can do this on your own. Let's see how you did. So 36% of the bases are adenine, and always adenine should be opposite of thymine. So approximately 36% of that DNA molecule should also be thymine. So 36 plus 36 is 72% of the molecule that you now know is A and T. So therefore, 100% of the molecule minus what you already know leaves 28 left. So there's 28% divided between the remaining two bases, G and C. So 28% divided by 2 means that there's approximately 14% of that molecule that is cytosine and 14% that is guanine. You should be able to do this for the upcoming quizzes and tests, by the way. Here's another practice example. If 15% of a DNA molecule is cytosine, can you calculate the percentage of the other bases? Pause the video and try this out on your own. Let's see how you did. Well, 15% C also means 50% G, so that's 30% of the DNA molecule that you've now accounted for. 70% of that DNA molecule is left, divided by 2 gives you 35% divided equally between A and T. So at this point in DNA history, scientists know that DNA is the molecule that stores genetic information. We know a little bit about nucleotide structure, phosphate, sugar, base, and we know that there's a basic shape. And this shape they know is a helix. It's a twisted helix of strands. Some scientists hypothesized that this DNA molecule contained 3 strands twisted together in a helix. Some scientists believe that it was 2 strands twisted together in a helix. So we need to figure out which one is correct. And so the following scientists are exploring that question. Oh, there we go. Okay, so Watson and Crick are these 2 scientists in Britain, and they were using ball and stick models to try to problem solve. So they were looking at all of the data from various different scientists using mathematical models and equations to see if they could figure out the chemical relationships between all of these atoms throughout the molecule to determine the structure and shape. So one way you can remember who these scientists were was Watson and Crick ball and stick models. So use that little rhyme as a clue. And in 1952, Erwin Chargaff shared his research about the base pair rule with them to help them kind of add more information to their equation. So they started to recognize, well, based on Erwin Chargaff's data, they hypothesized that the nitrogen bases must be arranged together on the interior of the molecule and the sugar and phosphates are on the outside. They were predicting that DNA was composed of 3 helices wrapped together. So they were building ball and stick models, trying to figure out if this is how the molecule actually is shaped. That brings us to our next team of scientists. Our next team of scientists are Maurice Wilkins and Rosalind Franklin. And I should use the word team here loosely. In comparison to Watson and Crick, who worked really closely together, like they worked together day by day in the lab, supporting each other, exploring together. In comparison, Maurice Wilkins and Rosalind Franklin, even though they worked in the same general lab team, they actually did not get along very well, and they did not work together. But they both used X-rays to photograph DNA strands. So they were both doing independent work in the same college, in the same lab, but they weren't necessarily working together because they did not get along. And what they found is that when you shine X-rays at a biological molecule, so here's a molecule, and you shine X-rays at it, what you see are the invisible X-rays bounce off of the atoms to create kind of a shadow where there is an atom present. So this creates a complex pattern in the photograph, which gives clues about the overall shape of that molecule. Here's that kind of broken down. Rosalind recognized from her photo 51 that DNA helix is made of 2 strands. So this is Rosalind Franklin's photograph. She took hundreds of photographs throughout her career, and this was talked a little bit about in the documentary you watched. But she extracted DNA, shown on an X-ray laser at it, and basically, again, where you see atoms within this molecule, it creates a shadow. And this photograph in particular was the most important for her because it had the most clear image. And this is photo 51. It's her famous photograph. Here's another way to look at this. So here are the backbones, and here you can see the base pairs. And so what you see here are the base pairs on each of those parts of the ladder showing up. And so what we can see here is we see that there's 2 strands. If there were 3 strands, there would be a third line or band of these shadows, but there are only 2. So in Rosalind Franklin's photograph, she was also able to use complex mathematical models to calculate the distance between each of these base pairs. So here's a base pair, and she was able to calculate the difference in size between these. And within her lab notebooks, her brilliance can be revealed. So even before Watson and Crick finalized their model, Rosalind Franklin had realized that DNA had a double helix structure, and that the way the component nucleotides or bases on each strand were connected meant that the 2 strands were complementary, enabling the molecule to replicate and copy itself. Above all, she noted that an infinite variety of nucleotide sequences, A's, T's, C's, and G's, would be possible to explain the biological specificity of DNA, which in other words means, remember how scientists used to think that proteins were the genetic molecule because there's so many different types and they're so complex? Well, what she discovered, or what she explained in her writing, was that based on the sequence of A's, T's, C's, and G's, there's an infinite number of ways that those could be added together in sequences to create the vast diversity of life that we see. So thereby showing that she had glimpsed the most decisive secret of DNA. The sequence of bases contains the genetic code that makes each of us similar but also unique. So solving the puzzle. Here's what's really tricky about this story. So Rosalind Franklin was doing her research independently. Part of research involves, conducting experiments, typing up your data, analyzing your data, and publishing it to share it with the world. And before she had a chance to do that, Maurice Wilkins took her photograph, this famous photograph, and Maurice Wilkins gave this photograph to Francis Watson, or James Watson and Francis Crick, without Rosalind's permission. And with this photograph, James Watson and Francis Crick were able to realize their mistake in their triple helix model, and they were essentially able to develop their own ball and stick model. So they constructed an accurate double helix model of the DNA structure, and they did not tell Rosalind Franklin that they got that information from her. Here's what they concluded. So here's the conclusion or the summary of DNA structure. The sugar phosphate backbone, which is phosphate sugar on opposite sides. This part is held together by strong covalent bonds. So you've got covalent bonds that hold all of these parts together along the sides. In the middle, you have nitrogen base pairs. And in between these nitrogen base pairs of each of the strands, there are very weak hydrogen bonds. So say, for example, this is a guanine and this might be an cytosine. These weak hydrogen bonds hold them together. What's cool about this and what's significant and important about it is that these weak hydrogen bonds can be broken, which means you can essentially rip this molecule apart in half. If you have an A over here and a T over here, that means you can just plug in the missing bases for each, which essentially allows this to unzip. And during DNA replication, we can therefore build two identical DNA helixes from one original template. Furthermore, each molecule, each strand of DNA can contain billions of A's, T's, C's, and G's up and down in a wide variety of different sequences to create the diversity of life that we see. The two strands are complementary and anti-parallel. They run in opposite directions. One strand runs in the direction called 5' to 3'. Remember that the 5' carbon is close to the phosphate, so that side is where you've got the phosphate pointing up. In comparison, the opposite strand will be the opposite direction, where the 3' end is up and the 5' end is pointing down, so the phosphate is pointing down. So you can tell that by looking here. So you've got the 3' right here, you've got the 5' over here, and the molecule is cut off here. But I'll show you in another image here. So again, here we've got the 5' phosphate pointing up, you've got the sugar pointing up, and then on the opposite side, you've got the sugar pointing down, and you've got the phosphate pointing down. So 5' end is down here, 5' end is up here on opposite strands. All right, so again, one strand runs 5' to 3', 5' to 3', and the opposite strand runs 3' to 5'. All right, so the four scientists all agreed to publish their research together in the same Nature journal in 1953. Watson and Crick published their article was first in the sequence of the articles published, and Maurice Wilkins and Rosalind Franklin's were in order after, and they basically provided the supporting data to support their theory. Rosalind Franklin's research in X-rays were vital to the development of an accurate model of the structure of DNA. But unfortunately, her research and contribution were overlooked, and at the time she was not recognized for her contribution. And in fact, what's really tragic here is that, especially in the 1950s and 1960s, women were not necessarily treated equally in science or in society in general. And Rosalind Franklin unfortunately fought an uphill battle to be recognized for her contributions to science. James Watson in particular vilified her and was pretty misogynistic in the way that he talked about her and wrote about her in his work. And it's really quite tragic. We'll be watching a little video coming up, but what's even worse is that in 1958, Rosalind Franklin actually died from cancer. She died from ovarian cancer, and it's possible that her cancer might have even been caused by the research she was doing, because remember, she was working with X-rays, and this was prior to them knowing that X-rays caused DNA mutations and possible cancer. So she was exposed to hundreds and hundreds of hours of radiation from X-rays, and it quite possibly might have been the cause of her ovarian cancer. And shortly after her death, in 1962, Wilkins, Watson, and Crick received the Nobel Prize for their discovery of DNA structure, and Rosalind Franklin was still not recognized for her role and contribution to their theory of DNA structure. So we're going to watch a little video. Please watch and add some notes to your notes as we go.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD. And she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab-mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind Elsie Franklin, the unsung mother of the double helix.
All right, so let's summarize. The scientists we've learned about, starting all the way back from the experiments to now, have built upon the knowledge that they gained from their predecessors before them. And by doing so, they were able to develop and provide the evidence needed to support the theory that DNA is the inherited material of cells and to understand its structure. One of my favorite quotes that I feel is very relevant to science is that one by Isaac Newton, which says, "If I have seen a little further, it is by standing on the shoulders of giants," essentially meaning we don't know what we know today without having learned from the people before us. So summarize your responses to the following questions. Number one, what is the relationship between the structure of DNA and its function? For example, how does DNA hold information relating to the bases, and how does its structure allow for it to be copied easily? Two, how have these four scientists contributed to our understanding of the function and structure? So summarize what each scientist did. Three, why wasn't Rosalind Franklin recognized for her contributions at the time? And four, what does the history of the discovery of DNA tell us about the nature of science? Thinking back to that Isaac Newton quote. All right, go ahead and respond to these questions to summarize what you've learned today. Thanks for watching. Send me a message if you have any questions. Otherwise, have an awesome rest of your day. Bye.